Hello everyone, today we're going to talk more about how we purify lactate dehydrogenase from bovine or cow heart. So by the end of this lab, we're going to be able to calculate the relative activity, total activity, and percent recovery for LDH during each step of the purification process. And so that's how we can track how effective our purification is after each step. Based on the purification table, again, you're going to evaluate the effectiveness of this particular step that you're going to perform during this lab, which is the ammonium sulfate precipitation. You're going to need to be able to describe the relationship between a protein's hydrophilicity and the concentration of ammonium sulfate at which that protein salts out. And then finally, you need to explain why after the ammonium sulfate precipitation, we need to dialyze our sample into a low salt buffer, and why this is especially important for ion exchange chromatography, or IEX, which is the next step of our purification. So I really like to start with a story whenever I can. And so one of the big questions for this lab is, why are we starting with a cow heart? Any of you who were hanging around this building during last semester when the cow heart was prepped last time knows that it's not necessarily a pretty process. And one reason is that starting with a cow heart is a lot easier for you guys and also for the TAs than the traditional method, such as overexpressing our protein in E. coli, which is what is commonly used in a research lab, because it requires less out of lab time for the TAs, myself, and for you. But the other reason is for historical reasons. And that's because back in the day, before we knew how to manipulate microorganisms like E. coli to express proteins for us, you had to find the tissue of an animal that overexpressed one type of particular protein and then extract the protein from that animal tissue. And one particular example of this is in biochemistry, you have or you will be learning about bovine pancreas RNAs. And a lot of early protein work where we learned about the structure and function of proteins through the work of one notable man, as an example, Christian Anfinsen, is because in the 1950s, the Armour meatpacking company had excessive amounts of bovine pancreas, and they didn't know what to do with it because people don't traditionally want to eat pancreas. And so they teamed up with E.J. Cohn at Harvard, and they found a way to roughly or crudely purify RNase H from the pancreas and then sold it to chemists at a very discounted price. And so a lot of early protein science work was done from large amounts of unwanted animal organs. And so in a spirit of early protein chemistry, we also are going to be purifying our enzyme from an animal organ. So part of it is because of ease and convenience, at least convenience for you guys, so you don't have to come in at weird hours to get your cells ready, but also for the sake of history. All right, so I'm mentioning protein purification here, but a lot of you guys probably haven't had any experience purifying protein yet, and you're wondering, what does this mean? Well, the goal of protein purification is to separate your protein from all of the proteins that are in the sample. Because when I give you a cow heart, so like here, this is what we're starting out with, you don't just get a pure sample of LDH hanging out in that heart. That protein is contained inside of a cell, and along with LDH in the cells, there's a lot of other proteins. And so we use a multi-step purification process to separate LDH from all the other proteins that are contained within a heart. So we have to break the cells open, and then we can use a series of columns, and purification methods, such as ammonium sulfate precipitation, which we'll talk about today, ion exchange chromatography, and then affinity, until finally, after we run our sample over so many different types of columns and through different types of purifications, that at the end, the only protein we have left is our protein of interest. So, the different characteristics that we take advantage of are the hydrophilicity. You can also think of this as the hydrophobicity. or the polarity of the protein, the overall polarity of the protein. And we take advantage of this property using ammonium sulfate precipitation, or ammonium sulfate cuts. And that's what we're going to be talking about today. You can exploit a protein based on its charge. And so that would be done via ion exchange chromatography. And that's lab 4. We can also take advantage of a protein via its like a specific binding characteristic. So like, I guess we'll just say binding here, but it's more complicated than that. And this is affinity chromatography. And that's lab 5. We can also take advantage of a protein based on its size. And that would be through size exclusion chromatography. And I'm going to put that as lab 7, but an asterisk, because we're going to be using size exclusion chromatography, but we're going to be using it to look at the size and quaternary structure of LDH rather than purifying it from the other proteins. So in a research lab, you might use it to separate it based on size, but we're using it for a different reason. And we'll talk about more of that for the video before lab 7. Okay, so I mentioned that I mentioned that our purification process is going to be multi-step. And so you want to know after each step, again, I mentioned we have to figure out whether or not a purification is effective and which steps are the most effective and which steps are the least effective. So in theory, next time when you go back and purify LDH, you can do a better job or make a better recommendation for the next class. And so we have to keep track of how much enzyme and how pure that enzyme is after each step of the purification. And the way that we can track this is through the purification table. And so you may create a new purification table for every lab entry you have, but you want to keep a master one where you put your final values, and then you'll use that in your final paper. Okay, and so when we have our purification table, we want to start with our crude lysate. And your TAs have kept some crude lysate for you. And the crude lysate is when you just break up the heart and see what's there. And so every step that purification step you perform, so the ammonium sulfate precipitation, lab 3, IEX lab 4, and affinity chromatography lab 5, you can go back and compare your values to what we started with when the heart was initially broken open. So in the purification table initially, and this is what you'll include as you purify in labs 3 to 5, you're going to add more later, but we're not going to worry about that right now. You're going to keep track of the activity, the dilution factor, so that's the amount that you have to dilute your sample before you put it in the activity assay. Then you want to give the relative activity in unit per mil, so this is the units per mil of your stock solution. And then we're going to measure the volume of each step, because then that will give you the total activity. So that'll give you the total amount of enzyme in your stock. So that turns it from a concentration into a value. And then finally, what you're going to do to have an idea for how much enzyme you have in each step, or how much enzyme is lost after each step of the purification, you're going to calculate your percent recovery. And your percent recovery compares your total units after each step, and you compare it back to the total activity and the crude lysate, so which would be the total activity that your TA started with when they started the purification process for you. All right, so now let's go ahead and go over these calculations. So the first new calculation that we're going over is the total activity. And the total activity is simply equal to the relative activity, which you learned how to calculate last week, times the volume in milliliters. And for the volume in milliliters, you're simply going to measure that using a graduated cylinder. And again, when you do your graduated cylinder, you want to make sure that the value that you're measuring is at the bottom of the meniscus of the liquid. And so the way that what I want you to do to get practice for calculating the total activity, which is again, gives us the amount of LDH in our stock, so our whole sample. What I want you to take a minute and I want you to use the relative activity and the total activity to calculate the total activity for the crude lysate. Go ahead, pause the video, and then I'll show you how to do the calculation. Okay, here we go. So total activity, TA, I'm going to abbreviate it, is equal to the relative activity times the volume. So our relative activity for our crude lysate is 67 units per mil. And then we multiply that by the volume of the crude lysate, which is 15 mils. Our mils cancel, and then we get, let me quick whip out my calculator here. All right, we get 1,005 units. And the amount of units that you have for your sample should be around 1,000. Oh, whoops, that's 10,005. My bad there. The amount of units that you should have around 1,000. Plus or minus like 500 units. All right? If it's not there, if you get 100 or if you get 10,000, your calculations are probably off for your actual sample. So come talk to me or to your the TA. All right. So the next calculation that we're going to practice is the percent recovery. So the percent recovery tells us how much sample or how much LDH we retain at each step. Or you can look at it, it gives us an idea of how much LDH we lose for each step. And so the way we calculate this is by taking 100, so that's where the percent comes in, times the total activity of the current step times the total starting activity. So for our case, that's going to be the crude lysate. So now take a minute and pause. And I want you to calculate the percent recovery for the IEX step. All right. So for the percent recovery of the IEX step, we have 100 times the total activity of the current step. So that's 850. 850 units divided by 1,005 units for the crude lysate. The units cancel. All right, and so we get a percent recovery of 84.5%. All right, and so the way that you're going to keep up your purification table with each lab as we attempt a different purification method, and this will be for this lab, for lab 4 and lab 5, you have to measure, you are going to take your sample before and after each purification step. You're going to figure out your dilution, and then you're going to measure the relative activity just as you did in lab 2. Then you're going to take your whole sample before you apply it to the column or throw in the ammonium sulfate or whatever you're going to do, and you're going to measure the volume of the sample in milliliters using a graduated cylinder. So you get the RA, and then when you measure the milliliters, you get the volume, but then you're also going to get the total activity. And then once you've completed, you've measured your relative activity, you have your volume of your sample, you're going to calculate the total activity and the percent recovery of LDH activity. And you're going to need to measure the volume in milliliters before and after each step, because as you take your sample off the column, your volume is going to change. So you need to make sure you take note. Your volume before and after a purification step is going to change. And then finally, this is super important, and that's for each purification step before and after. So pre 65% an ammonium sulfate cut, and then post, you need to save approximately, I think it's like 300 to 500 microliters. I can't remember exactly what the value is, but your lab notebook or the lab handout will tell you. You need to save a couple hundred microliters of each sample before and after the column and purification step. And the reason for that is because later on you're going to calculate some other factors to expand upon your protein table. We're not worried about those calculations now. We just need to make sure you're prepared. and you give those samples to Lindsay or whoever your TA is before you leave, and then you'll come back and use them in Lab 8. Okay, so now that we've kind of got an overview for the calculations and the overall purification process, I want to talk to you more about the science that we're going to be performing today, as well as the science that your TAs performed before you got to lab in order to get your sample to where it was before you could actually work with it. So what happened? So first things first, your instructors went out to a slaughterhouse out in Yadkinville, and they purchased this cow heart. Funny story, I went with my family to Publix over Christmas, and my mom goes, "Ew, what's that?" And I look at it, and I go, "Oh, that's a cow heart." And I knew because I had prepared the cow heart for fall of 2017. Anyway, so you get this big honking cow heart. Your TAs will cut it up into chunks, and they're like chunks that they should be like an inch cube. They throw it into a blender around with some neutral pH buffer, and they blend it up, and they make this lovely, fragrant pink cow heart smoothie. And so you grind up the heart in the smoothie. And I guess that goes for down there too. And so the reason that we use heart is because it's a muscle and it contains lots of LDH. And so we have a high amount, we pick a tissue that has a high amount of our protein so we can get a good yield, so you have lots of units to purify over the course of the semester. Because trust me, you're going to lose a lot of enzyme over the course of the semester. So once we homogenize, we grind up the heart in a blender, we get this pink concoction. And the result is it's kind of like orange juice, and that you have the LDH goes into the liquid, but then you still have a pulp left over. And you have to separate the liquid from the pulp because the LDH is located in the cytosol, so when your TAs break open the heart cells, the LDH spills out, but you still have all the tissue. And so the technique that your TAs employ to separate the LDH that's in the liquid or the supernatant, as we call it as biochemists, the supernatant, they can use centrifugation, which separates particles based on their density. And so what happens is if we start off with a cell homogenate down here, so that's the LDH and other proteins and liquid plus the pulp. So to be accurate, we would draw lots of polka dots that are floating around solution there. That would definitely be a cloudy pink solution. When we spin the homogenate at 20,000 times G, that means that your supernatant stays, and this is going to be a bright red color. Whereas the cell debris, including, the nuclei, and then the cell debris, as well as any mitochondria, peroxisomes, and lysosomes, so those are organelles in the heart cells, are going to collect in a pellet at the bottom, whereas the supernatant that contains LDH and other proteins is going to be up here. So when we spin our homogenate, we get LDH and the supernatant, LDH and other things, and then we get cell debris in the pellet. And so what happens is we can decant the super, and so we get LDH and other things, and then we leave the pulp behind. Because you don't want to put pulp on a purification column. Trust me, I have done it before, and it's not on accident, and it's not pretty. And so you'll see here that the speeds are referred to as times G, whereas you might be used to seeing motor speeds and RPM. And so we refer to, whenever you see a purification protocol, it's usually going to be written in times G, and that's because the RPM is related to the size of the rotor. And so what happens is if you have is that if we have the JA21 102, so over here, this rotor is smaller, whereas the JA20 108 is larger. The RPM, an RPM of, for instance, of 500 RPM is going to translate into a faster speed, a faster times G, when you have a bigger rotor than a smaller rotor. And so we do all of our calculations in times G because in order to make sure that you spin it at the correct G force, you can adjust the RPM depending on the size of the rotor. And so usually with centrifuges, you can switch the setting to times G and it'll do that math automatically for you. But in those cases, like the centrifuge that I used in graduate school that was actually older than me, yes, there were lots of equipment in my lab that was older than me. The centrifuges at that time didn't have that capability, so we had to actually have a table like this one over our centrifuge so we knew which speed to spin it at. But anyway, yes. So remember, LDH is in this bright red supernatant, and then the pellet is where all the cell crud is, or cell debris. I guess that's a nicer way to put it. So then after your TAs are left with a supernatant, and it's bright red, with LDH, the next step becomes, okay, we have LDH, we also have all these other proteins. So how do we separate LDH from all these other proteins? And that's where the different purification steps come in. And in our lab, the steps in this particular lab, the steps that you're going to be taking is you're going to separate based on hydrophilicity. That's lab 3. Then we're going to separate based on charge. Lab 4, I know I'm repeating myself a bit here, but repetition can't be too bad. And then affinity for lab 5. So today, we're going to talk about how we use an ammonium sulfate precipitation to separate proteins based on their hydrophilicity. You can also think of it as their hydrophobicity or their polarity. But I'm going to explain it in terms of hydrophilicity because that's the easiest for me to wrap my head around and explain in the most effective way. So in our, let's say in our solution, we have a hydrophilic protein. So this would be a protein that has lots of hydrophilic and polar amino acids. So amino acids, they're going to have alcohol groups, carbonyls, they're going to end in those kinds of groups. So these are hydrophilic amino acids they're going to have. And charges. So these are going to be your charged and polar amino acids. They're going to have a lot of those. And then, so that's our purple guy. And then let's say we have a not very hydrophilic protein. So these are going to be proteins that don't have a lot of hydrophilic amino acids. These guys are probably going to have a higher content of aromatic and greasy. So going to have proteins with side chains like that, leucine. They're going to have rings. So those are going to be your greasier proteins. And then this guy here represents a water molecule, and then this guy here represents an ammonium sulfate ion. So let's say we're hanging out in a solution where there's just water, and we have our hydrophilic protein, our hydrophilic, and our hydrophobic protein over here. So when we just have these proteins hanging out in water and maybe a little bit of salt, both these proteins are happy because what happens is the water molecules are going and they're solvating the hydrophilic groups on both of these proteins. Now, the hydrophilic protein has more residues, so you're going to have more water molecules solvating that protein, whereas the hydrophobic protein is going to have fewer hydrophilic groups that are going to be, so it's going to have fewer water molecules that are surrounding it. So again, if we have like a serine, you can see how the water molecules could be like hydrogen bonding and having polar interactions with those groups. Okay, so everyone's happy when we don't have any ammonium sulfate. However, what happens is when we start adding ammonium sulfate, things start to act a little fishy. And you can see here that the ammonium sulfate ions start interacting with the water molecules instead of the hydrophilic groups on the protein surface, because it turns out that it's a lot easier for water molecules to solvate a really tiny ammonium sulfate ion than it is for them to solvate the large, bulky groups of a protein. So for our hydrophilic protein, so Phil over here, some of the water molecules that are solvating its surface are going to get scavenged to solvate the ammonium sulfate ions instead, but there's still a bunch of hydrophilic groups there that are being solvated by waters. So at a low ammonium sulfate concentration, Phil is pretty happy. He's still solvated. He's still hanging out, and he's hanging out in solution. However, Phoebe over here didn't have a lot of hydrophilic groups to begin with, and so when you add just a little bit of ammonium sulfate, the ammonium sulfate is going to scavenge all the water molecules from the surface of Phoebe over here. And so what happens is when all the molecules are taken away, it's easier for the protein to fold upon itself and to have those hydrophobic, that's a really pathetic phenyl group there, sorry guys. It's going to be easier for those hydrophobic groups within the protein and the hydrophilic groups too to glom upon themselves than it is for the protein to try to scavenge and interact with the salt that's in solution. And so what happens is once all the water is taken away from the protein will aggregate and will actually form a solid or a precipitate, and it will fall out of solution. So then let's say we add even more ammonium sulfate. So we have a higher concentration. It doesn't make a difference for Phoebe over here because he has already crashed out of solution. But for Phil, eventually we're going to hit a point. It's going to take a lot more ammonium sulfate to take away all the water ions from the surface of the protein. But eventually we're going to hit a point where all those water molecules are going to be exhausted. And then Phil too is going to resort to glomming upon himself because it's more favorable to do so, and then fall into in form of pellet as well. But for Phil, that's going to take a lot more ammonium sulfate than it did for Phoebe over here. All right, and so one of the ways we can quantify and we can figure out how much ammonium sulfate we can use to get a protein to pop out of solution or to stay in solution is we talk about the percent ammonium sulfate. And so what this means is that at 20 degrees, and for our calculations, it says in the handout it's 0, but we're actually doing our calculations at 20, and that at 20 degrees Celsius, it takes 536 grams of ammonium sulfate, so we're going to abbreviate that AS, to saturate 1,000 liters of water. And so what that means is so let's say we have 536 gram per liter ammonium sulfate per solution. Once you add 1 more gram, so you take into 537, it's not going to dissolve because you've saturated the solution. And so if we have a 10% ammonium sulfate solution, that means that it's 10% of 536 grams per liter. So 10% ammonium sulfate solution would mean we have 53.6 grams ammonium sulfate per liter. All right. And so what happens is that different proteins, depending on their primary sequence or the way that once they fold and where the different hydrophobic and hydrophilic proteins are arranged, are they in a patch, are they evenly distributed, all that's going to affect how much ammonium sulfate it takes to induce a protein to fall out of solution. And so for many proteins, this has been experimentally determined. So somebody long before us went ahead and did the dirty work for us and figured out at what percentage of ammonium sulfate LDH came out of solution. And this is a fairly crude purification technique, and it's usually done in 2 to 3 steps. But we can use the hydrophilicity or the percentage at which we know our protein falls out of solution to manipulate it and to get the minimum amount of proteins in the pellet. So we can figure, and that's why we use 2 steps. So we try to minimize the number of other proteins in our pellet. So so your TAs first did a 45% cut, and that's because they wanted to get rid of the hydrophobic, so the really hydrophobic junk proteins. So those proteins that we don't want, we do a preliminary step because we just want to get rid of all the really hydrophobic stuff. So you're cutting out a whole bunch of proteins right out of the gate. And then the 65% cut, so we know that 65% is where LDH is LDH's precipitation point. And so we do a 65% cut so we can get LDH. All right, so thinking about this, I want you to take a minute. So here's a table where we have a protein and we have the point, the percentage of ammonium sulfate at which that protein precipitates or goes into a pellet. So again, talking about those 2 cuts, we have our 45% cut and our 65% cut. This is the TAs, this is you in class. So I want you to pause and I want you to predict and draw where each protein would be in the cut. And then you can play again and I'll walk you through where it is. Okay, so for a 45% ammonium sulfate cut, if we look at our table here, so for 45%, any proteins that precipitate at less than 45% are going to be in the pellet. Any proteins that are going to be greater than 45% are going to be in the solution. So we can see here right off the bat, protein A precipitates at 25% ammonium sulfate, so that's going to be protein A. But and also protein B has a precipitation point of 40%. So protein B is also going to be located in the pellet. However, LDH's is at 65% and protein C is at 90%. So the supernatant is going to be LDH and protein C. By the way, these other proteins are hypothetical proteins. There are many more proteins in your real solution. So after the 45% cut, we're going to throw out the pellet. So that goes away, and then we're just left with the supernatant. All right. So at the 45% cut, so at the 45%, we have all the proteins. But at the 65%, when we do the 65, we're only going to have LDH and B. So once we have the 65% ammonium sulfate cut, LDH precipitates at 65%. So LDH is going to be in this pellet, whereas protein C comes out at 90. So protein C is still going to be hanging out in solution. And so at this point, we would take our pellet and then we would resuspend it in buffer. All right. And so you can see here that based on the 45% cut, with that first cut, we get rid of 2 crap proteins that we don't want right off the bat. And then the next step is where we more fine-tune and we target our protein of interest. So that's why normally ammonium sulfate cuts are in 2 steps. So then after we take our protein, we resuspend it in 30 millimolar bicene, pH 8.5. And bicene is non-ionic. So it doesn't have a salt. It's an organic, I think it's one of the goods buffers. And so we resuspend that, and then we have to dialyze. And so the goal of the dialysis is to get rid of the ammonium sulfate, or we'll just say the AS here, the ammonium sulfate. So once we want to get rid of all that ammonium sulfate, and we want to have a nice low salt buffered solution. And so the way that we do that is we take a dialysis membrane. So I've drawn a picture here. And so the volume of our dialysis, this is again also going to be 30 millimolar bicene, pH 8.5. And it's going to be 2 liters. And so then in your solution, you've resuspended it in 20 or 30 millimolar bicene, but you're still going to have some ammonium sulfate in there. And so we have to get rid of the rest of that ammonium sulfate, or get rid of as much of it as we can. We'll talk about why in a minute. And so the way that we do that is we put that small volume of our protein plus ammonium sulfate into a dialysis bag. And this dialysis membrane, it has holes that are small enough, so if we have our protein here, and we have salt as X's, it's small enough that salt can get in and out of the membrane, but protein cannot. And so what happened then is, over time, the bicene that's in the buffer and then the ammonium sulfate are going to equilibrate. They're going to go in and out of the membrane, so they reach an equilibrium, but your protein is going to be caught in the membrane. And so as a result, if we have the bicene that's the same concentration inside and outside of the membrane, which is exactly what we do, we make our dialysis buffer on the outside the same as the resuspension buffer, that's going to change the same. That's going to stay the same. But you're going to find that the ammonium sulfate is going to leave the bag and go into the solution, so it equilibrates, and the result is that the concentration of the ammonium sulfate in the dialysis bag is going to be diluted. And we can figure out the extent to which it's diluted using the M1V1 equation. And so if we have an example here, all right, where let's say we resuspend our pellet in 20 mils. Say we resuspend our pellet in 20 mils of let's say it's the same bicene buffer, but it has 100 millimolar NaCl, and we're dialyzing against 2 liters of 30 millimolar bicene, pH 8.5. What is the final concentration of NaCl in the dialyzed sample? And then we're not going to calculate the concentration of bicene because it's going to stay the same. So if we start off, so our initial is the volume of our dialysis bag. So we have 20 mils of 150 millimolar NaCl. So this is our bag before we put it into the dialysis buffer. However, we take the dialysis bag, we put it into the buffer, and we allow it to equilibrate. Our volume becomes 20 mils of the bag plus 2,000 mils. So that's 2,020. So that's the volume of the bag plus the volume of the dialysis buffer. And then we don't know what the final concentration is, because again, we're effectively diluting it by making the volume of the salt larger, but by keeping the volume of the protein the same. And so we can solve, if you want to pause this, but I guess I've already gone ahead and worked it. So we can take, so that would be 20 times 150 divided by 2,020. And that's going to give us a concentration of 1.5 millimolar NaCl. So you can see that by taking our sample and putting it into 2 liters of bicene buffer that has no salt, that we've diluted our sample 100-fold. And that's a lot better. And then you may be begging the next question, which is, why do we go through all the trouble of dialysis? Why don't we just take our ammonium sulfate sample or our resuspended bicene that has some salt in it and then not worry about it? And that's because the next step because lab 4 is ion exchange. And so we're doing anion exchange chromatography. And this is because at pH 8, LDH has a negative charge. We'll talk about the logic behind that more in detail later. So what that means is we're going to take a positively charged column and then flow LDH over it. And what happens is if we have ammonium sulfate masking the charge of LDH, LDH is not going to stick. It's going to go straight through, and so we wouldn't be able to effectively purify the protein. So we need to make sure that we get rid of the salt so the charge of LDH isn't masked by the ammonium sulfate, so it can stick to the column and then be eluted appropriately. But again, we'll talk more about that in detail during the next lab. So now I'd like to do a quick review of the experimental details, so what happens before lab and what you're going to be doing in lab, and then I'll also go through the steps for solving another buffer problem, since we can never do too many buffer problems. All right, so the first step is homogenation. So it says here the butcher, but in reality, your instructors are going to go to the slaughterhouse and pick up a fresh heart, and they take the heart muscle, they remove the fat, and then chop it up into 1-inch cubes. They add some buffer, so at a neutral pH, and blend it up. So you break up the cell walls and then release the cytoplasm into a buffered solution. So AKA, we're going to release the LDH and some other proteins into the buffer. Then we take the sample, we spin it down to 20,000 times G, so we get the pulp and the organelles in the pellet, and then LDH and all the other soluble proteins in the supernatant. Yes, and this is a beautiful red solution, like beautiful ruby red. Then your Ts will go and they'll take it and they'll add ammonium sulfate slowly to make a 40% saturated solution. So again, when we do ammonium sulfate, we want to keep the solution cold. We want to add slowly, because it turns out that when ammonium sulfate doesn't like to dissolve into cold solutions, so you have to add it slowly, and this is anywhere from 4 to 6 batches. And then finally, you want to minimize bubbles, because having bubbles in your solution can actually cause your protein to aggregate before you reach the precipitation point. So again, after this, the sample was centrifuged to separate the aggregated protein pellet from the supernatant. At this point, the percent ammonium sulfate is less than 65%, which is LDH. So LDH is still soluble, so it's in the supernatant, which is kept. We'll dispose of the pellet, and then that sample was frozen at -80 until it was time for you guys to use it. This black bar means it's time for lab. And so what happens at the beginning of lab, you're going to be given 100 mils of the saturated ammonium sulfate solution. So you're going to measure the volume to make sure it's 100 mils. Maybe you have 101, maybe you have 98, fine. You're going to measure the volume, and then you're going to run an activity assay to determine how many units are present at the start of the purification. Then you're going to take out 500 microliters. You label with your initials and then the purification step. So for me, this would be AGB, and this would be post, I guess this would be, post 40%. Then you're going to freeze it in an Eppendorf tube for analysis later. Then you're going to launch into your 65% ammonium sulfate calculation. So we already did the math for you to determine that you need to add 1.166 grams of ammonium sulfate for every mil of solution you have. So you can do the math based on your volume to figure out how much ammonium sulfate you need to weigh out. Then you're going to put your solution in a beaker, you're going to surround it with some ice on a stir plate, and then you're going to keep it stirring. Then you'll slowly add ammonium sulfate, so 4 to 5 or like 6 additions over 10 to 20 minutes. And then you'll notice, again, you'll see some cloudiness happening, and this will be LDH among some other proteins that are precipitating into solution as you add the ammonium sulfate. Then you're going to take your sample, you're going to pour it into 2, I think it's 2, or 1. It'll be 1, and you'll balance against another group into a centrifuge tube. We're going to run upstairs. We're going to spin it down to 20,000 times G. And in theory, your LDH should be in the pellet, and there should be none in the supernatant. However, before you move on to the next step, you need to check. And so you'll resolubilize your pellet in the 30 millimolar lysine at pH 8, but then you have to measure the volume and the supernatant, measure the volume and the activity assay of both the supernatant and the pellet, because you have to make sure that LDH activity is in the pellet. Because if not, then you have to go back and repeat the precipitation, but with more ammonium sulfate. So it's of utmost important. So that's why we have to go back and we have to double check, and we have to make sure that LDH comes out in that step. So then once you can confirm that LDH is in the supernatant, you're going to take out a 500 microliter aliquot. You're going to label with your name and or your initials, so AGB for me, and the purification step, and then you'll freeze a little bit in the Eppendorf for you to use in lab 5 or lab 8. And then finally, once you've performed the ammonium sulfate cut, again, we need to get rid of that salt, and we need to get the pH right for the next purification step. So our TA again is Lindsay. So Lindsay and I will help you to resuspend that pellet, to transfer it into the dialysis tubing, and then overnight we're going to exchange it into 4 liters of 30 millimolar lysine. And again, we need to be really careful with this step because it's really easy for you to lose LDH at this step. I know of at least one person last semester that spilled their dialysis bag and we had to do a benchtop extraction. So just be aware, and remember, it's really important, don't throw anything out without asking Lindsay or me first. And so finally, at the end of lab, before you leave, I want you to have your purification table started, and Lindsay or myself needs to look at your purification table before you leave. So you're going to have your crude homogenate, your 40% super, your 65% super, and then your 65 resuspended pellet, and you'll have determined the activity, the dilution factor, so how you diluted your stock enzyme before you put it into the activity assay, as well as the relative activity, the volume in milliliters, the total activity, and the percent recovery. And so we can take a look at that, and then you can ask any questions before you leave. It's also an opportunity for us to make sure that you're performing the calculations correctly. And so with that, I'm going to end this video, and I'll post the supplemental video on solving the pH problem in a separate video because they're already at 47 minutes. All right, good luck.